Thanks, Masood. Hi, everyone. I realize it's a strange audience. The live video is going to 3 people that I already know, 4 people that I already know, actually know what I'm talking about already, so it's fine. But I assume we're going to get into a little bit more of this stuff, or at least we'll get some more viewership on the YouTube video later. Masood, are you willing to share that PowerPoint presentation? While Masood is getting that ready, I'll just say that this is really just a very brief whirlwind tour of some of the work that is taking place in our lab at the University of the Western Cape. And really the objective is to give people a feel for the kind of work that we're doing, and the hope is at least that we can get some Q&A type things going at a later stage. But let's see how that goes. For now, I'll just run through some of the ideas of projects that are currently taking place in the lab and stuff that I'm currently interested in. I think it's really important to remember that the work that is taking place in the lab is the vast majority of that work is taking place, or is taking place thanks to others, to students and other researchers. So I just wanted to point out a few of the students that are currently in the lab and whose work I'm going to touch on in various aspects today. Several of you know Robin, who is a research fellow in the lab, and she does a lot of the work on particularly trophic ecology. Then Jody Barnes, who's done various work that I'll talk about a bit later in Kruger, and is moving on now to work on desert peltasts, egg eater feeding. And then a slew of master's students, really, including Celinda Kuhle, Elam, Brian, Kim, Janine, Matthew, Tilo, and new to the lab is Ryan. And Ryan is listening in here, hiding behind the name of Melissa Petford. But also there's a lot of other research that takes place. A lot of the work has taken place at the hands of undergrad and grad students who have now left the lab, and also some of the work with other collaborators. So when I give this kind of presentation to other people, I think it's really important to give people a background into what snakes really are. And I think probably the most important thing for people to realize is that it's very easy when talking about reptile biology to differentiate out snakes and lizards. But really it's important to remember that snakes are just this strange group of derived lizards. Snakes are really just weird lizards. And they have got some features that make them really, really special. And the thing that really jumps out at me and is really key to a whole lot of the discussions that we have in the lab is that the fact that snakes are really just a lineage of over 3,000 species in which every single species is a predator. And so that means that predation biology and the biology of feeding on other animals is really, really important in the biology of snakes in general. So in that context, though, it's easy to think of snakes as being this monolithic thing, kind of just, this is what snakes do. But it's really important to realize the remarkable diversity that fits within that. And so this is a slide that I stole from another presentation, and it highlights some of the sort of broad groups of snakes. And these are just the sort of families that are known from Southern Africa. And you can see that we get everything that ranges from the first 2 on the left, which are sort of burrowing snakes that mostly live underground and almost exclusively eat invertebrates and their eggs, all the way through giant pythonids, venomous vipers, which are really familiar to many people around the world, a range of colubrids, including some medically important ones like that vine snake in that picture, and then obviously things like elapids, and a huge slew of diversity of a lineage that is diversified in Africa, the Lamprophiids, and a range of other families that are sometimes included in Lamprophiidae or sometimes not, like Samophines. But importantly, they encapsulate a huge quantity of diversity, not only in the habitats that they occupy, but also in their feeding and how they interact with organisms within ecosystems. Next. So when I joined the University of the Western Cape, I was really tasked with, basically I had to write my first research grant application, and the thing that sat with me, or the questions and the thought processes that went through my mind, arose from a discussion I was having with a friend at the time. And what that friend was saying to me was, are snakes even important in ecosystems at all? It's very easy to to flippantly say, oh, snakes are really important in ecosystems because they do X, Y, and Z. But are they really important in ecosystems? And it got me thinking, if you want to conserve snakes, you probably have to demonstrate some degree of what they're doing in ecosystems. And two, I thought about, well, if I was actually going to go ahead and try and understand the importance of snakes in ecosystems, or at least the roles of snakes in ecosystems, that I'd need to differentiate the kind of research focus into three main elements. And those are briefly listed over here. And the first is how are snake and really reptile communities in general, how are they structured? In other words, which species co-occur together where? Really, this is going to fit under the label of community ecology. The second element that I was really interested in, and all three of these were largely lacking for many African systems, was how abundant are snakes in these different communities. And what we're getting at here are the different demographic processes that are driving what's going on in snake populations. And the reason these are all important is that if you want to tell how important a snake is or a species of snake is in a particular ecosystem, you need to know which species are actually there, in other words, what that community is made up of. But you also need to know how abundant those individual species are, because it's that abundance in that ecosystem that is likely to dictate the magnitude of the effect that they're having within that ecosystem. Species that are really rare tend to have relatively small effects. Species that are really abundant often have really profound effects in ecosystems. And lastly, the element that I think really links to the idea of snakes as predators is this idea of what do snakes eat. I've spoken with many of you before about this in many contexts, but really this comes down to understanding snake trophic ecology. We know that all species of snakes are predators. One could argue that snakes have their biggest impact on ecosystems either through their role as predators, directly as predators, or even indirectly as through things like the impacts of landscape of fear. So controlling how other organisms in that ecosystem are acting through that risk of predation. Next. So the first element of this and how these link it up to projects in the lab are related to how is trying to answer how are reptile communities actually structured. And so what that means is that we have several projects within the lab, some of them formal, some of them relatively informal, that relate to trying to understand which species of reptiles make up the communities, and particularly which communities of snakes co-occur in different landscapes across southern Africa. What that involves is quite a lot of animal sampling, including stuff like trapping, and you can see a photograph from one of our reptile diversity and African savanna field courses with a bunch of students learning how to install a drift fence to capture snakes and lizards and a bunch of other small vertebrates. But we also spend a fair amount of attention on things like ad hoc observations of where different species occur. And we've also started utilizing and driving quite a lot of the citizen science elements, including things like iNaturalist in particular. But in all cases, or nearly all cases, and especially for the snakes, the biggest challenge that we face probably relates to just incredibly low detection probability for so many species. For years, people have said, oh, if you go to Africa, or a lot of people think if you come to Africa, you're going to see lots and lots and lots of snakes in particular. But it turns out that snakes are actually quite difficult to see in Africa. Many people who visit here are surprised, possibly because they're used to these really, really diverse and really numerical or really high abundance snake populations in parts of the US, Europe, and Asia. But it turns when you come to Africa, often finding these snakes and encountering them is incredibly challenging. And it's those low detection probabilities that really have an implication for how you sample communities, because it means that you have to sample communities and patches where organisms potentially occur. You need to put in incredible survey effort to make sure that you're sampling entire communities, and that has huge implications for how we actually go about doing these surveys and how much effort we put into them. That's not to say that we only have to rely on actually detecting animals at particular places. And some of the work that Jody Barnes did for his master's showed that actually with a clean data set, you can start making modeled predictions of where species occur using classical species distribution modeling, but then also that you can start making some inferences about what those communities of those organisms look like by starting to sum those model outputs in that. So a few results. The figure that you can see over there is the Kruger National Park. For those of you who don't know, the Kruger National Park is a protected area in the northeastern part of South Africa. To give some of our American listeners context, it's relatively large. It's about 20,000 square kilometers. On a relative scale, think sort of twice the size of Yellowstone. And it's relatively well sampled for reptiles, or at least of all of the protected areas in Southern Africa, at least anything approaching that scale, we have a pretty good understanding about which species occur there. There's at least 3 books that have been written explicitly about the reptiles that occur in the park and that. And despite all of that information, what Jody found out was that the actual data that support these distribution maps and the records of different species in different places in the park are not nearly as thorough as we thought. So you can see he went and mapped out all the known actual localities available, and he managed to, after removing duplicates and triplicates and all of the rubbish and some of the erroneous records, he ended up synthesizing it down to about 7,500 records that are mapped on the left there. And importantly, if you start looking at those, you can see that those dots are not randomly dispersed across the park. And in fact, they're biased towards where there is human infrastructure. That's what that figure at the top right is suggesting. And you can use these records, even though they are relatively sparsely sampled, and especially if you have information about biases in their sampling, to make predictions about the distribution of reptiles within this context. So if you look at the bottom right-hand side, the bottom right-hand corner there, what you can see are 3 different distribution maps. And these are the modeled predictions for 3 species of reptile that occur in the park. And they really range from really quite weak predictions. The first one on the left-hand side, where that's nearly all yellow and green. And you can see that the model support for that is relatively low. Conversely, you can see some species where we've got relatively good support, and it seems to match habitat preferences quite well. So, for example, that middle species is Nile water monitor. They're strongly aquatic, and you can see a pattern that is really pulling out all of the river systems as being key habitats for those fine-scale distributions. And then as a last example, you can see a species of gecko, and you can see where it's relatively restricted to the northern parts of the park. Jody took it a step further, though, and he started taking those models and those model projections, including, unfortunately, some of the species that don't perform, where the models don't perform particularly well. But it turns out if you start overlaying those, what you start to get are reptile communities, or at least predictions of reptile communities that come out at very broad scales that set up a whole bunch of hypotheses for people to test. Now, the reason that this is important is if you're going to monitor reptiles and reptile communities in a conservation or biodiversity management context, you need to have some sort of mechanism to compare your measures to. There's no point in telling people, oh, we want to monitor reptile populations, and so we'll go and we'll do an active search once a year in a good season or 3 times a year in appropriate seasons. You can't do that when the fact remains that several of the species that occur in this area are occurring with such low detection probabilities that they only get seen once every decade or 2. It's not to mean it's not to say that they're disappearing from those areas. It's just that they're going extended periods without being detected, even with survey effort. And so this is really a step towards coming up with theoretically predicted hypotheses for what those reptile communities can look like, and starting to provide a way forward to start monitoring reptile diversity in the park. The next part is that was really the reptile communities and how we start thinking about reptile communities. This next little element is quite short, and it relates mostly to snake demography. Basically, it starts with us trying to understand how abundant different species can be in ecosystems. And a little bit of a result that I've got here for you is some of Selinda Kuhle-Tokota's work. And this is really interesting in that he was interested in trying to find out why these 2 species of snakes, that's a spotted scarp sterker, Samophylax rhombiatus, and a crossmark whip snake, Samophis crucifer. He was really interested in finding out how they actually partition their respective niches, because they seem to co-occur. They're similar body size, they have similar diets, They seem to co-occur throughout most of their global distribution. And in fact, in some habitats, they seem to be the two most abundant species there, at least of snakes where we work. And so he set out to try and understand, are they partitioning their ecological space by occupying different parts of the landscape at relatively fine scales? And although this work is still very preliminary, what we see is that even at relatively fine spatial scales, what we're seeing is a broad positive correlation between the abundance of one species and the abundance of the other. What that suggests is that you get some areas that are just low snake abundance sites and some areas that are just high snake abundance sites. And that in fact these two co-occurring species seem to be co-occurring at similar densities across this gradient. And so there has to be a different element within a different dimension at least, within their niches that they might be partitioning. It probably relates to diet, but it's not 100% clear because they definitely have diets that do overlap and they do share prey resources. We also, beyond just sort of the traditional mark recapture type things, we've also taken a few steps towards trying to understand patterns in demographic processes for things like puff adders. This is on the back of a huge mark recapture, or at least a huge radiotelemetry data set, predominantly collected by Xavier Glaudas, while he was busy with his postdoc with Graham Alexander, and then to a lesser degree myself. And what we've been trying to do there is really utilize those radiotelemetry data to start estimating sex and season-specific differential mortality rates in puff adders. And this really links to the idea that despite them being puff adders, being large-bodied venomous species, it turns out that in many parts of their distribution, they experience just remarkably high natural mortality rates. It seems as though, as a broad guess, it seems as though an adult puff adder in South Africa is experiencing approximately a 50% chance every year of dying. So annual survival rates of somewhere around 50%. For context, if you compare that to some species of rattlesnakes in the US, those estimates in the US often range, for similar sized organisms, range from anywhere from like 70, high 60s, all the way up through 80 and 90% annual survival rates. Okay, the last element that the lab works on relates to trophic ecology. As I mentioned, it's mostly what are snakes feeding on, to try and answer that part of the equation of, we know which snakes occur in a region, we know how abundant they can be in some cases. So what are their actual effect on other organisms? And that happens through their feeding ability. Now, the way that we do this involves a bunch of different approaches. A lot of it includes very traditional examination of museum stomach contents, stomach contents of preserved museum specimens. That picture on the top left is me smiling at a Cape cobra that had been dissected, and we found a gerbil inside of its gut. It turns out that specimen was collected over 100 years ago, and to have access to a trophic interaction that took place more than 100 years before your time is really quite an experience. And several of the students, past and present, have relied quite heavily on examining museum data to better understand the feeding ecology of snake species. We also are trying some other techniques, including trying to sequence fecal remains of certain snakes, particularly those scarpstekers and crossmark whip snakes that I spoke to you about earlier. And so that's one of the students in the lab currently. Kim is working quite hard at trying to develop tools that will allow us to go out and really in detail sample the dietary records that are coming out of those populations to understand that niche trade-off that I was talking about. And then, as many of you know, we also have been heavily involved in utilizing the citizen science through social media as a mechanism for gathering trophic records. Mostly this takes place through Facebook, through a group that we call Predation Records for short. Excuse me, and really the figure that I want to show you here is on the bottom right. And it's a bar chart showing the cumulative number of diet records for the whole of Southern Africa for the last 100 and something years. And importantly, what you can see there in gray, the gray bars, not the blue bars, the gray bars show a sort of an unsurprising increase in the number of records as time has progressed. And in fact, if you look closely around the mid-60s, you can see the line does a little bit of a jump. That was when Don Broadley published his PhD. And then you can see a climb from the late 90s through the early to mid-2000s. That was associated with Rick Shine deciding to visit Southern Africa and spending a huge quantity of time with several local collaborators just dissecting museum specimens. So that resulted in a slew of papers coming out. Importantly, though, in terms of the social media stuff, you'll see that the rate at which we're now gathering records, in other words, the rate at which those blue bars are stacking up, is faster than we've ever collected records in the history of African herpetology. And by way of example, in the last 5 years, or in the 5 years that we've been running predation records, we've gathered about the equivalent of the last 110 years worth of record we've gathered in 5 years now. And so just a remarkable increase in the rate at which we're gathering highly detailed feeding records for snakes across Southern Africa. And it really shows the scope that citizen science has to answer some of these questions. It's the trophic ecology stuff that seems to have been the source of most of the academic work that has come out of the lab. This is just to show you some of that trophic ecology stuff and give you some examples of the kinds of papers that have been published. You can see they range all the way from trying to better understand things like ophiophagy and cannibalism in cobras and in vine snakes, specifically in 2 examples here, through broad dietary studies, just like trying to better understand Cape cobra diets, and then also trying to, how do I put this? Trying to pitch dietary studies in interesting evolutionary contexts, such as the Journal of Zoology paper down at the bottom. And then the last element of work that we do, that I know that a lot of people are really interested in some of this work, mostly because of the adventure involved in it, relates to the work that Robin, myself, and several collaborators are doing in the Kalahari at the moment. For those not from South Africa, the Kalahari is a desert region in the sort of central western parts of the country. And it is this remarkable landscape that is occupied by a range of cool organisms. And I'll explain as we go through some of these slides why we think they're cool and some of the experiences that we've had in trying to study them. So the driver of this entire system are these huge sociable weaver nests that occur throughout the Kalahari. So the sociable weaver is that little passerine bird that you see on the left of your screen. It's about the size of a sparrow. And importantly, what these birds do is they build these giant communal thatched colonies within isolated mostly these big acacia trees throughout the Kalahari landscape. Now, an individual tree might have as many as 500 or 600 chambers in it, and they're constantly breaking down, building, doing extra stuff and that. But many of these trees host these colonies for decades on end. And there's now research showing that the actual colonies themselves and the resources that the birds are bringing have influences on the growth of the trees and in features of the trees and how the trees grow in that. The little bird that you see on the right-hand side is a pygmy falcon. Pygmy falcons co-occur in these systems, and in fact, they're obligatory nest parasites. And so they utilize these things, occasionally eating the little weavers that utilize the social weavers that utilize the nest. Importantly, though, sociable weavers are ecosystem engineers, so they build these huge structures, but their movement and their foraging in the landscape has huge implications for how other organisms utilize that landscape. So, for example, there's research that shows that they're bringing in nutrients towards the base of the tree from anywhere as far as 4 or 5 kilometers away. It turns out that large mammals are utilizing trees that have these nests in far more frequently than they utilize other trees. It turns out that the antelope will often aggregate under these trees for shade, and so you get increased defecation. You get a whole bunch of ecosystem effects that sprawl out from having these trees in the landscape. Now, you might say, well, Brian, why are you studying birds? But the real interesting thing is that these bird populations are pretty much the single most important factor. You can go back one. The single most important factor that dictates these bird population sizes is predation by snakes. And so what you can see in this image here, the top right is a Cape cobra and the bottom is a male worm slug. And these are snakes that have crawled up into chambers having just fed on a whole bunch of sociable weaver chicks and eggs and are sitting there waiting, digesting those meals out, and we've located them there. So what we try to do is understand the role that these snake predations have on driving this entire Kalahari ecosystem and the ecosystem engineering effects that these birds are having. You think about it this way, the birds are dictating huge elements that are taking place within the landscape, and it turns out that the snakes are regulating bird populations. To give you an idea of just how important that effect can be, it turns out that if a breeding colony of birds has a breeding attempt and they might produce 50, 60, 70 eggs or something like that across multiple chambers in a single attempt, it's not unusual for a single snake to visit the colony and to go through systematically and eat every single egg and or chick in the nest. And so we can have snakes that completely prevent an entire colony from raising a single chick in an entire breeding season. And that happens relatively often. So it's really a dramatic effect. And the way that we study them is quite a lot of walking around and just spending our time looking up into sociable weaver nests, occasionally getting lucky with those images that you can see over there. But we also do stuff like radio telemetry, and you can see Robin over there dialing in the radio frequency for a radio transmitter that's implanted into one of our Cape Cobras. And we get to move around this really beautiful landscape studying Cape Cobras in a wild part of Africa, which is really fantastic. It's not that working in these African systems is boring, ever. And I like to include this when I'm talking to audiences that are not familiar with South Africa. This is just a little anecdote of some fun that we had the one time. In the top image, you can see me holding the radio telemetry gear. And if you look down the road beyond me, what you should see is a pack of African wild dogs running down the road towards us. Now, I don't need to, or many people might think that that's terrifying. It turns out that there's no confirmed records ever of an attack by African wild dogs on humans. However, if you've ever seen what a pack of wild dogs can do to an animal much larger than a human, you'll understand why we responded the way we did. And so what you can see in the next few images there, we had been tracking a snake in a tree. It was hiding in a sociable weaver colony quite high up in a large acacia tree. And what we had to do is myself and Graham and Robin, the three of us, eventually climbed up the tree somewhat embarrassingly while the dogs gathered around the base of the tree and looked up at us as if to say, what are you guys doing? You know, this is so pathetic. And when we eventually told, I mean, the dogs eventually ran off a few minutes later, but when we told some of our colleagues that work there, they just laughed at us saying, it's outrageous that you've had to do that. They've literally had situations where they've been able to walk up to feeding wild dogs and pull carcasses away from them, and the dogs do nothing. But all the time that we get to spend in the field is not just spent running away from African wild dogs. We also get to spend a lot of time with radio telemetered snakes and just with untelemetered snakes that we get to we encounter in the field. And from that perspective, we've got to see some really interesting biology play out. So these are two separate images. Both of them did quite well on social media, so several of you might have seen them. The top image is a male Cape Cobra cannibalizing a smaller male Cape Cobra. And our work has shown now that, in fact, if you look at the consumption of other Cape Cobras by Cape Cobras, that it's really quite abundant. By way of example, if you go through the entire list of known prey records for all Cape Cobras that we have in our database, and it's now at about 107 individual feeding records, it turns out that puff adders are the most abundant prey item, most abundant snake prey item today. Snakes make up about 40% of all records. Puff adders are the most abundant snake prey item, and the next most abundant snake prey item are the Cape Cobras, before any of the other things like mole snakes and some of the other snakes that are quite abundant in the landscape. In that context, though, we've also had observations of cobras hunting puff adders, and to say that this is a simple thing would be just completely wrong. The photograph that we have over there, Robin took the bottom image there, and that's of a cobra that we ended up spending basically 2 days with as this battle royale played out, with the cobra finding the puff adder, biting it, the puff adder trying to get away, the cobra retreating, and then the puff adder lying on the surface feeling the effects of venom, shortly thereafter, the cobra would come back, bite again, and then disappear again. This went on for days, and we got to witness this entire procedure before the cobra eventually was able to swallow that very large meal pup adder. So that's just a brief rundown of some of the stuff that we do in the lab. Any of those papers that I mentioned are available on the lab website. Feel free to pop on there and grab them. And really, at this point, I just want to take any questions. Thank you, Masood.
Yeah, looks good to me. Bryan, thanks for a really interesting talk. It's kind of like looking into my own brain. I knew we had a lot in common before now, but I never realized quite how much. So yeah, really.
I must tell you, Andrew, that when Robin met you at that meeting, she came back and she said, "You have to talk to Andrew Dosa. You guys are exactly the same."
That's really funny. Yeah, it was such a pleasure to meet her in Snowbird, and I'm sorry that we didn't meet. I think you were in New Mexico at the Biology of the Vipers meeting or something at the same time. So
no, I had to come back to teach.
That's right. Okay. Yeah, in fact, I made a note to myself here. I should really think about trying to come visit your lab for maybe my sabbatical in several years. But we should talk. I mean, I don't want to monopolize. If other people have questions, I have a few, but
well, you might as well start with one before anyone else asks.
Sure. So, so you mentioned, first of all, so thinking about like top-down effects that snakes have on their prey, I think my thought process followed a very similar trajectory to yours, which is that we always use that as a justification for why people shouldn't kill snakes or why they're important in ecosystems, why we should have snake conservation. But there are actually very few, almost no data whatsoever showing that they do have important top-down or any effects in ecosystems. I mean, I believe that they do, but that's not science, right? So I've sort of collected this list of papers that seem to suggest it or include some data on top-down effects that snakes might have on their prey. A lot of them come from systems where the snakes are not native to that system, right? So that's like the most obvious way to manipulate it. But it sounds to me like your sociable weaver system is just the best one I've ever heard about for an opportunity for the snakes to drive the ecosystem engineer that's driving the landscape. So, and I should say also, so I visited Southern Africa. I visited Namibia for the first time last year, and we saw these sociable weavers and pygmy falcons and no snakes, like you said. I mean, it's pretty disappointing, but a lot of people prepared me for that. So, you described these boomslangs or cobras going into these sociable weaver nests and wiping out every egg and every juvenile. What kind of time period does that occur over? I mean, how can they possibly eat so much at once? They're not blind snakes, but it reminds me of what you read about a blind snake going into a termite mound and gorging on hundreds of larvae or something.
So we've spoken about this a lot and thought about this a lot. The way that they are doing it, so for one, Kalahari Cape Cobras, and particularly the ones that we're finding in nests, are large-bodied snakes. So it's not atypical for us to find a snake of 1.7 meters or so in size.
Okay. They can eat quite a few eggs.
Yeah, exactly. And a sociable weaver egg is a small thing. So a sociable weaver egg is typically about 2.4 grams. And so you can fit a lot of 2.4 gram eggs into these relatively big, robust cobras. A lot of people have this idea that active foragers are, because they seem to, on average, eat smaller meals, that they're not capable of still fitting in a huge quantity of food inside of them. But if you think about that puff adder that I showed at the end there, that was a 680 gram snake, and it ended up inside of a cobra in that. So you can fit a lot of eggs into a 680 gram meal if you can get it down. And because they're small, as you say, much like Scalexophidians or some of the sea snakes, what they're really doing is they're actually going and browsing almost from these nests. But it's happening quickly because a single, let's call it raid on a nest might be taking place in a period of half a day. For a small nest, it can take place in a matter of an hour or so. It turns out they're not very good climbers, and so that's the one thing that the weavers have going for them, is that cobras often fall out. Well, they can access the edge colonies quite well, but they tend to fall out trying to get to the central colonies. But yeah, they can do it, and we've had, because we interact quite a lot with the ornithologists that work in these systems,
Sure.
and they've had cases where birds or individual colonies of birds have attempted to breed 3 or 4 times during the season, and every single time they've had a snake come in and take out the entire brood.
Wow. I mean, that's massive, and I haven't gotten into the literature on sociable weavers at all. I mean, the colonial aspect really makes them a lot like an ant or a termite. I wonder, probably ornithologists know what their genetics are like and what they sort of choose to do, like how the groups make decisions about what to do when the nest colony itself is raided repeatedly or is under attack. Can they move? It's a huge investment for them to build a new nest in a new tree somewhere. So I guess they're not abandoning that.
They stay with those nests, and so the behavior of the birds during a raid and the response of the birds through a season of raids is really interesting. Well, no, the behavior of the birds during a raid is really interesting. Their response is to just breed again. That's all they do. They breed again, breed again, breed again. And these trees have been there for years, and some of these colonies have unambiguously been there for decades. And that was one of the things that drew my attention to the system in the first place, because it's very rare in snake ecology to have a prey resource that is simultaneously temporally predictable, because they breed in very predictable responses to rainfall. You get a certain threshold of rainfall, the birds start breeding, snakes visit the colonies. But this resource is not only temporally predictable, but it's also spatially explicit, because it's not like I go to a general field and hope that I can catch a mouse that's running around, and maybe if there's more mice I can catch something. This is a case of, if it rains, I go in 10 days to this exact tree that I've been to before, and there will be food there.
Yeah.
So it's a fascinating system. But when an actual snake arrives at a colony, the responses of the birds varies. If the snake is not inside a chamber yet, so if it's approaching a chamber or crawling around the outside, the birds will make a huge noise. They'll try and flush the snake away in that. Importantly, male birds make a bigger noise and a bigger show to try and just fend off the snake if there are female birds present to watch them making the scene.
Wow.
If there are no female birds present, the effort that the males make is greatly reduced, which is great. The next thing is, the moment that snake climbs into a colony, so into one of those holes, all the birds except the birds who occupied that chamber stop responding, and they carry on going about their business. It's as though the snake isn't there at all. So the one bird or the 2 birds that might be occupying that chamber will keep disappearing, like keep making a scene and trying to get in, trying to chase the snake away, but all the other birds in the colonies just stop. And so they really do have this benign neglect approach to being raided by snakes. They make a bit of a scene. They're not really good at fending off the snakes. In fact, one of the hypotheses for why the sociable weavers tolerate having the pygmy falcons nest in their nest, even though they occasionally eat weavers, one of the hypotheses is that the pygmy falcons keep snakes away. They don't. Really? The snakes eat pygmy falcon chicks as well. But there is recorded evidence of pygmy falcons actually fending off snakes from raiding a nest.
Wow. So why aren't there more snakes? I mean, what's limiting the populations of those snakes?
I've been thinking about this for about 10 years now, and I don't have a good answer for you yet.
Mm-hmm.
I jump between one of 2 things. One, there are as many snakes, but there are just a whole bunch of reasons why in a predator-rich ecosystem you should be difficult to be found. Mm-hmm. Option 2 is, what constitutes normal quantities of snakes is dictated by places like the US and parts of Europe, and that where we've got fundamentally shifted ecosystems that might have artificially high representation of certain species. Mm-hmm. all the way through to magic. I don't know.
Yeah, it's really the central thing that I think limits snake population and community ecology, which you touched on so nicely at the beginning, is that it's just impossible to do any population or community ecology when you can't properly count them, and when you can't even properly detect whether or not the species are present there. That's the same thing that has been stymieing my research program since it began. And it's so interesting, like, how can they be so abundant in some cases but be so damn difficult to detect?
Yeah. So to give you an idea, we've got 200 artificial cover objects out at that Scarpstierker crossmark whipsnake study site. Mm-hmm. We've been checking those 200 cover objects about once a month, at times more frequently, at times less frequently, but about once a month for the last 5 years. And we still don't have sufficiently high recapture rates to get any of the traditional mark recapture models to tell us anything informative.
Yeah, I'd say that's typical, honestly. And you're right, there are systems, particularly North American and European systems, that are well documented where that level of effort might just barely be sufficient to get a mark recapture model to converge. It might not. And yeah, like you, I've racked my brain about, well, how do we do this better, right? I mean, you can use a better method. I think for terrestrial snakes, there probably aren't any better methods. For aquatic snakes, some traps, I think, work better than an artificial cover object. You can use dogs, you can use eDNA, you can use citizen science in these huge, databases, but then you have to think about all these other problems that you touched on even earlier when you were talking about the Kruger reptile surveys. How do you do a species distribution model when you don't know what the biases in sampling are, especially if those data sets have been gathered opportunistically by many people over decades, and most of those people didn't understand the importance of recording when they didn't see a species. You just don't have much trust in them, do you?
No. And so, I think that finding a better way of doing it, as you said, is something that's driven me for a long time. The way it currently looks to me is that we either need to shift some of the questions in how, basically reset the target of what we're actually hoping we can actually achieve within snake ecology. And so over this lockdown, I've been thinking quite a lot about just looking at relative detection rates across transformation gradients, so habitat A, habitat B, with enough sampling in each of those. You don't need to make sure that you've got the entire community, but if I consistently try to sample that particular species across that particular habitat transformation, and I consistently get a negative, a positive, or a neutral effect, I can start then telling about how communities might respond without sampling the entire community. This is obviously not nearly enough, but it's better than what we currently have.
Yeah, I think you're right about asking different questions. I mean, my master's work, I also used stable isotopes to try and get at questions about snake trophic ecology. And what I came to realize is that asking the question of what do they eat may not be the right question, right? Maybe we want to know more about, like, how much does the diet of species A overlap with that of species B? Or how much does the diet of this species change over time or differ between sexes? Those are maybe more answerable questions than this vague, like, very intuitive, but possibly impossible to answer question about snakes of what do they eat.
Absolutely. Masood, you look like you have a question.
Andrew asked a couple of questions that I had written to ask you. And on the weaver, it is really amazing. I have seen videos of how they build those nests, and it is really eye-popping the way that those little birds are working. And a few things that you said was totally new to me on that one. You mentioned impact on the trees, and I don't know whether from that viewpoint, whether the snake has a negative impact on that, as opposed to the birds. That's one thing that I wanted to you to touch on. The other thing was that I assumed totally I assumed that most of the perdition was taking place by boom slangs, and you keep mentioning the cobras, and I said, whoa. So there may be other ones as well, because I have seen another one with another species of a snake in there that I actually posted to the group. But aside from that, so cobras are the main predators, or boom slangs are main?
So that's a really important point. The area that we work in the southern Kalahari represents a relatively small overlap between Cape Cobra distribution, which is an arid western distribution, and boomslang, which is a savanna species. And the Kalahari is really this arid savanna ecosystem. And so the area where these sociable weavers occur, at least in South Africa, is right on the edge of boomslang distribution. So there are, in parts of the range, boomslang are the most important predator, but then as you move further west, boomslang disappear out of the ecosystem, and Cape cobras appear to have access to the system uninterrupted or without any competition from boomslang. At both of our sites, boomslang are present, as are Cape cobras, but Cape cobras seem to be in higher abundances here. You remember, you're right on the edge of boomslang distribution. And so that actually raises a whole bunch of other questions about how boomslang cope in this ecosystem, because it turns out that when the birds don't breed, and getting into a whole background of foraging ecology, the boomslang are relatively restricted diets. They tend to only eat birds, eggs or chicks, and then some species of lizards, as those prey items account for about 95% of all prey items. And they particularly eat chameleons. When I say lizards, I mostly mean chameleons. And in the Kalahari, chameleons are not very abundant at all. And the birds that are breeding are almost all breeding in response to rain. And so you go through these bouts where it doesn't rain for extended periods of time, where there's zero bird breeding activity. And when you find a boomslang in that situation, their body condition is absolutely trashed. So we've had 1.7 meter snakes, 1.7 meter boomslang that have weighed as little as 400 grams. So like tiny skin and bone things. And when the birds breed again, they bulk up into these big fat sausages. The cobras, on the other hand, will eat literally anything. I think that if you sort of left your stray dog outside and it was small enough, the cobra would eat it. And what this means is, though, through these lean periods where the birds don't breed, I think the cobras are able to shift into other prey like snakes or small mammals or whatever they can get into their mouths. And it's during these periods where there's no real competition from boomslang, the boomslang populations start crashing off when they can't get any food. The cobras are able to just classically shift to a different prey type. And so that's why the cobras are able to tolerate existing further west. And that's why they occur together in some places, but not entirely in others. In terms of your question about how the influences of the snakes on the trees is a really interesting one. One of the things that happens in these ecosystems is that those nests get so big and so massive that if you get bouts of rain coming through, there are extended periods where you get any waterlogging in those masses, in the nests. The nest becomes so heavy that they fall off and break branches off of those trees. So in many cases, it's the aggregation of all of this mass on these branches that results in these catastrophic branch breaks in these trees. So in many cases, snakes suppressing colony growth reduces colony size, or at least keeps colony size below thresholds where if they were left unsuppressed, they would break branches off. As evidence for this, the team from UCT that works on the sociable weavers at one of the sites that we work on, the sociable weavers are obviously fascinating for a whole bunch of social biology reasons. They have helper breeding systems and things like that. But they have these students coming from around the world to try and study helper breeding and breeding events of these birds. And they've had situations where a student comes out from another country or another continent spend some time trying to waiting for these birds to breed. The moment the birds breed, they get to the age where they start displaying all these interesting helper behaviors, and a snake comes into the colony and eats everyone. And so they've started excluding snakes from trees by wrapping the base of the tree in cellophane and like cling wrap, and it stops the snakes from being able to climb up because they don't have the traction. And it's not published. The effect isn't very strong because they have a relatively small sample size. But the data look like they suggest that if you keep snakes out of those systems, those colonies, the number of individuals in those colonies grow and the size of the colonies grow as a direct effect of that. So there are these very clear links between predation of the birds and what those birds are doing to those trees in a negative way. But it's complex because they're also providing extra nutrients for those trees. And we know that the nutrients that are being defecated by the birds based on the isotope data are being pulled up by the trees, are found in the leaves of the trees straight off, at some point in that. So it's this complex relationship that the birds have with the trees, and the snake predation kind of sits on top of all of that, occasionally pressing down the bird numbers.
It's a perfect example of snakes and ecosystem and birds and trees, and that's perfect.
Yeah. The other, if I can just, before you go on there, the other, and Andrew mentioned it as well, as systems where snakes might influence landscapes through other species. The other good example in southern Africa are egg eaters, Peltasts, these little egg-eating snakes. They eat a diet that is exclusively restricted to bird eggs. But in some ecosystems in southern Africa, they can occur at such high densities that they are taking just enormous quantities of eggs out of nests at certain times of the year, and as a result probably restricting those birds in performing their ecosystem services, things like seed dispersal and things like that, really being restricted through this effect of predation by the egg eaters.
The other question that I had was related to what you said about the natural causes of death for puff adders. And you mentioned something like 50%. That was eye-popping for me, that natural causes will cause that much anything. But could you explain a little bit more on that? What sort of natural causes we're talking about?
So the study site that we worked in is a nature reserve, so it has something that that approximates an intact ecosystem with all of its predators and that in it. Probably the most dominant, numerically the most common predator of puff adders, of radiotelemetid puff adders, were snouted cobras, which is the big species of non-spitting cobra that occurs there. Mammalian predators, things like honey badgers and blackback jackals, accounted for a fair number of fatalities. We have several species of specialized snake eagle, and we had several individuals that were taken out by snake eagles. We had incidental deaths that were natural but kind of weird. We had two separate occasions where ambushing puff adders that were ambushing on the edge of game trails got stepped on by giraffes, and that ended up smashing up their heads. Neither of those animals survived. And so just this huge diversity of things that could kill you as a puff adder, and they seem to do it. The interesting thing is puff adders definitely have, as Graham Alexander always says, a weedy biology. It turns out that puff adders have enormous litter sizes. They have live birth, and the record for any snake that gives live birth, number of babies in a single litter, is from a puff adder from East Africa, where they get quite a lot bigger. That was kept in I think it was the Prague Zoo. Somewhere in Europe, it was a captive animal, and it gave birth to 156 babies in a single birthing event. And that's obviously dramatic, but some of our smaller puff adders down here, it wouldn't be atypical for a puff adder female to reach sexual maturity in her third year of life. In that first reproductive year, she would have between 10 and 20 babies. There's a good chance she's going to skip her fourth year because it takes a while to recover resources, but we don't know that for certain. And in her second breeding attempt, she would have grown sufficiently large in her fifth year to have between 30 and 50 babies. And so they're producing just enormous numbers of babies into the landscape, maintaining these very large populations, but having just really, really strong top-down removals from those populations. It's an amazing system to think of how rapidly they're dying off. But the basic numbers that we know about their reproductive biology fits in. You can calculate the number of new babies that you need each season in order to sustain an adult population that's experiencing a 50% mortality rate, and the numbers balanced.
Yeah, that's insane. I had no idea they were so R-selected and fecund.
No, it's outrageous.
That's wild.
So the other southern African viper that we have sort of survival data for was the Namakwa dwarf-adder work that I did during my PhD. And that's a much, much smaller critter. It's a tiny little sand-associated viper. A really big female would be maybe 23 centimeters in total length. When they're born, they are about 10 or 12 centimeters long. A newborn snake weighs under 3 grams. They're tiny. And they also have annual mortality rates that, at least the population that I studied, the best estimates of population survival rates there annually was about somewhere between 48 and 56% annually. But they do it in a different way. Puff adders have these huge bodies and they can have lots of babies inside of it. But when you're really restricted in your body size, you're actually limited in the total amount of space that you can have for developing embryos. And so although dwarf adders only have sort of 4, 5, or 6 babies in a single breeding attempt, unlike most other vipers, Namakwa dwarf adders breed every single year. And they can do that because they live right on the coast. And so the effects of the ocean temperature regulate seasonality on the coast. And so you have a relatively aseasonal environment that they live in, and they have food availability, and they do feed throughout the entire year. So they feed throughout the entire year, maintain resources throughout the entire year, and it looks as though they're able to breed every single year in order to deal with this relatively large predation effect.
I was just going to ask if you found any evidence of Namakwa dwarf adders breeding more than once a year.
No, we haven't, mostly because of the way that we tried to do it is not very sensitive. So we looked at percentage of dissected or examined museum specimens that had reproductively active organs. And at most, it appears as though it's sitting at about 90, we think that probably 90% of the population is breeding every single year. But we don't have, I haven't looked into any captivity data whether people can get them to breed twice a year. But they're so small.
It's pretty rare in snakes. I mean, I've only seen maybe a few instances where there's a snake that's known to breed more than once a year, in contrast to lizards where like most of them or many of them can do that.
Exactly.
Yeah, it's interesting.
Well, doesn't it have to do anything with the resources? Is there enough prey for them to get strong? Because you just mentioned that they wait a year before giving another set of birth.
You're talking about puff adders now? Yeah, so the one thing that we don't have great measures on is how much food an individual puff adder is consuming and processing in a year, and how much food she, in this case, has access to. I don't doubt, we've seen females recover body condition really quickly in good seasons, such that they possibly could breed every year if, for example, you have rodent population booms. Rodents make up a really big portion of their diet, and I think that when those populations explode, they particularly take advantage of it. So we've seen telemetrid females post-partition recover condition in a matter of a few months, and that would suggest that they might be able to do it if resources are available, but we don't have really a good handle on how frequently that happens. We don't really know how those sorts of densities of rodents translates into what is enough food availability. And then we also don't have a great idea on how puff adders prey switch as different stuff becomes available, because we know that they also eat lizards and in some cases toads, and they also occasionally eat birds. And so measuring encounter rates with all of those things is quite complex. Xavier Gladas, again in Graham's lab, published an excellent paper on using fixed videography. So he actually put video cameras focused onto ambushing puff adders. And he has the closest data that we have to what an actual puff adder encounters in terms of feeding opportunities. But that doesn't really allow us to separate out what happens when background levels of abundance of rodents or increases or decreases in abundance of rodents. We just have this picture of that they are actually encountering a large variety of different things, and they will strike at and consume a large variety of these things. The last thing I'll say about Xavier's work, because it's fascinating, is that fixed videography also provided evidence for two separate types of prey luring. So he published a paper that showed that they caudal lure, so they wiggle their tails to attract predominantly lizard prey, I think it was. They also do this weird tail waving thing under certain conditions. We don't know why. But when they're approached by toads, they stick their tongue out and they waggle their tongue from side to side as lingual luring. So they have these two separate mechanisms for prey luring, which is spectacularly cool.
Yeah, I read the paper and it's really fascinating.
Yeah.
We have kept you a lot, but let me ask a final question, and that's on a snake bite. And since you're working with the communities, and snake bite is an interest of mine as well, how do you manage, or how do you work with the communities that you are working with in relation to the ecology, the need for the snakes to be there, and the dangers of the snake bite, and I'm assuming a lot of rural communities. So how are you managing those two?
Yeah. So several of the study sites that we work in are protected areas, and many protected areas in South Africa, rural communities have been removed from for a whole range of reasons that I don't think this is the right place to get into them. So several of our study sites, we don't interact with local communities that often. Our major interaction with people, with members of the public, is not really through rural communities, but through students, particularly undergrads, that we interact with at university. And the reason that that's the case is that the university that I teach at, the University of the Western Cape, predominantly serves students from disadvantaged communities. The vast majority of our student populace, for many of them, it's the first time that anyone in their family has attended tertiary education, so they're first-time college attendees, and many of them still have pretty rural backgrounds in many cases. Nearly all of them still have rural family homes and that. And so we spend a lot of time working, even within basic undergrad courses and that, talking to people about snakes, snake bite, snake ecology, and walking this fine line between snakes are really cool and you can learn a lot about them and they're really interesting, but countersetting that with, yes, where you live, and even in Cape Town, there are venomous species that you should absolutely not touch. We're not saying you should kill them, but you should absolutely not. No person who doesn't know what they're doing should be handling a Cape Cobra. It's as simple as that. It's a very, very dangerous animal. So we spend a lot of time with students trying to drive in the value of understanding organisms, even if those organisms are dangerous. Probably the biggest extension of that is this course that Robin and I run together with a few others called Reptile Diversity in African Savannas. And if you just do a search for, in fact, if you go to the website studyafricanreptiles.org, you'll see some of the field trips that we run there. And the objective there is to take students from universities that are predominantly being occupied by students from disadvantaged backgrounds, and to give them the opportunity to spend time in the field studying organisms in nature. So we take students who, in many cases, when they write their application, say things like, I'm afraid of snakes, but I want to come on this trip because it looks interesting. And by taking those people into wild places and talking to them about the biology of these organisms and showing them organisms that are not really easily accessible, like informative natural history about local snakes is not easily accessible. And in fact, the only real organization that provides any information like that is the organization that Ashley works for. But our students don't get to really interact with organisms, and so taking them into a wild place, showing them real animals, showing them the actual biology of those real animals, and showing them potentially very dangerous animals, but in a very controlled and safe manner, all sort of acts together to turn those people into, for lack of a better term, snake ambassadors. And so a lot of the students leave those courses and end up spending a lot of time talking to others and talking to family members and end up running their own programs and courses and that. So I think that's probably the biggest way in which we engage with the greater public about the sort of this dichotomy of snakes are cool, but by the way, some of them also kill people.
Great. If nobody else has any question, we'll let Bryan go. It's over to time. Thank you very, very much. I appreciate it.
Thank you, Masood. Thank you, everybody. I appreciate coming through.
Okay.
Thanks a lot, Bryan.
Have a good day, and again, appreciate it. Ashley
Thank you.
says it's a cool talk. Thanks. Okay. Bye-bye, everybody.
Cool.